It's time for Researchers Come. I am Nadi, interviewer. Today I'm in Awajishima in Japan to participate in Focus on Microscopy, the international conference. And today's researcher is Dr. Eduardo P MELO, the Institute for Biotechnology and Bioengineering. Nice to meet you, Dr. MELO. How do you feel, Japan?
Well, people in Japan are very kind, you know. It has been a very good experience to be here. I arrived 4 days ago. I'm planning to visit Kyoto and then and Tokyo. So I'm really happy to be here. It has been a big pleasure to visit Japan.
Okay. Thank you very much. So let's start today's interview. First of all, could you tell me your current research theme? I hear that your study is about the stability of proteins.
Exactly. So all my scientific career has been devoted to the study of protein stability, unfolding and folding pathways. So the study of protein stability is important from the biotechnological point of view, because if you want to make a product using, for instance, an enzyme, you want to keep the activity of that enzyme for a long time. So it's important to avoid the unfolding of the enzyme. Protein stability is also very important from the biomedical side, because you have a lot of diseases which are related to the misfolding of proteins, and so you can really apply your knowledge on protein stability with different focus.
Okay, thank you. So the three-dimensional conformation of protein is very important for their protein's function. Thank you very much. Could you tell me your approach, the research approach?
Yes, so to study what we have to do to study protein stability in vitro, what you have to do is you have to purify that protein. Then you use mostly biophysical techniques to study conformational changes of that protein. So I use mostly spectroscopic techniques like fluorescence, circular dichroism, light scattering. So all the techniques that allow you to watch the conformation of your protein.
Okay. So you observe the conformation of protein by using the biophysical technique.
Exactly.
Okay. Thank you very much. So what kind of research, what you can provide, offer to other researchers? Could you tell me?
So I can provide my knowledge. So if anyone has a protein and wants to study the stability and the unfolding of that protein, I could be helpful and collaborate with that people on the study of that stability.
So your research is mainly the enzyme, but your knowledge will be applied to any kind of proteins.
Yeah, that's true. I work mostly with enzymes, but my knowledge can be applied to any protein, so I can study the unfolding and the stability of any protein. Doesn't matter the function, since you are looking at structure.
Okay, thanks. I would like to ask you about your future research theme. What you will do in the future?
So recently, I have been devoted mostly to the study of prion diseases. I have done a sabbatical period with Adriano Aguzzi in Zurich, Switzerland, and since there, I want to study the mechanism of prion diseases, and this is related to what I have been doing so far, because prion diseases relate to a conformational change of a prion protein and to the misfolding of that protein, and then the aggregation process of that protein. So I'm really using my knowledge to study or to contribute to study the mechanism of prion diseases.
Prion disease is so-called mad cow disease, and it is caused by misfolding of the prion proteins. Thank you. Okay, could you tell me your approach to solve the prion disease?
Yes, so besides the biophysical approach, to study prion diseases with a more global approach, you need to enter the cell biology field, because the prion protein is attached to the cell surface. So if you want to study the conformational change of the prion protein in its cell microenvironment, you have to use a cell biology approach, imaging techniques, and so on. So that's why I move a little bit from the biophysical side to the cell biology side.
Thank you. Finally, I would like to ask about what you need from other researchers. Could you tell?
So it's very important to have very good equipment to run your research, and so it will be nice to collaborate with people having, for instance, a fluorescence confocal microscope, or so people related a little bit more to the cell biology and imaging techniques. It would be nice.
Okay, so thank you. I forgot I forgot to ask one more thing. Your motivation to your research. Why you are interested in this kind of research team?
When you are in research, it's not for money, for sure. It's so I think it's something that is, I like to acquire knowledge and to have knowledge and to understand the mechanism of life, and so I think this is the strong motivation to go on and to continue to follow my research lines.
Okay, thank you very much. So it's time. This is all of our interview today. Thank you very much. After this conference, I hear that you go to Kyoto or Tokyo.
Yes, so I will visit Kyoto tomorrow. I hope that you suggest me some nice places to see in Kyoto, and then I will spend the weekend in Tokyo.
Okay. So I'm living in Kyoto, so I will tell you where to visit or where to eat later.
Yes, thank you very much.
But I don't know about Tokyo, so please ask someone about Tokyo. Thank you very much. So it's time to finish. Today's researcher is Dr. Melo. Thank you very much.
Yes, thank you.
So see you next time. Bye bye.